Hello everyone, this is Dr. Chandrasekhar from Chandu Biology classes. In this session, I'm going to give a brief analysis of complete syllabus for APPSC assistant professor, especially for the biochemistry category. So for rest of the categories, we will do a separate sessions. And I'm going to do a complete analysis of the syllabus, how many number of units to be studied, what are the chapters that you can skip, what are the books to be followed, and when the coaching classes is going to be commenced. I will explain everything to all of you. Now focus. So since we are all having a limited time, and I am especially focusing on all the assistant professors who are already working in all the 18 universities in Andhra Pradesh. So as you also know very well, most of the assistant professors are having a very limited time. So by considering your timetable, we are starting a batch from the November 10th onwards, which is a 40 days bullet speed revision course. In this particular course, we are going to cover all the important topics which will help you to score very surely in the upcoming exam. Along with that, you will get chapter wise as well as a mock test series. Most importantly, you will get high quality study material for chapter wise as well as well crafted PowerPoints with the shortcuts where you have to focus especially towards the exam point of view. All will be provided to all of you once you enroll in this particular course. And second point, most of the aspirants and most of the assistant professors, after seeing the syllabus, most of them get panicked because the syllabus of biochemistry, biotechnology is especially huge. So what kind of a material that we have to follow? Everyone know very well, if you want to cover the biochemistry, you have to read multiple books. But do you really have a time to read multiple standard reference book like Leninger, White and Boyd, Strayer, for the cell biology, Alberts, immunology, QB? We have a huge syllabus. Now you also know very well, we do not have a time to read all these standard reference book. So by considering your situation, we have framed or we have developed a excellent study material which contains a total number of 6 books. This particular 6 books will cover all the 10 to 12 units which are mentioned in our biochemistry syllabus. So the book is going to be released from the 10th November onwards. And our revision course is also going to start from the 10th November. The timings, everything I'm going to inform you by tomorrow. That is on Monday. So in this particular session, let's focus on every pinpoint of the syllabus. Okay. Now let's start understanding about the syllabus. In this, I will explain to all of you how many number of units to be covered. Okay. And the second point, most importantly, we will focus on most important topics to be covered in every single unit, because we don't have a much time to cover every single topic because our time is limited and the syllabus is huge. And again, most of the students are having a doubt about the books. Okay, so now let's focus. I will solve all your queries in this particular session. I am requesting all the assistant professors who are working in the universities currently, requesting you to go through the complete video analysis. Then only you will get a crystal clear idea about how to do our preparation. Okay, now let's start. So biochemistry, according to the syllabus, there are 12 units were given. And the syllabus is really vast. I can say that among the entire APPSC syllabus, I can say that the biochemistry syllabus and biotechnology syllabus is completely vast. And remember, the assistant professors who are applying for the category of biochemistry, I am requesting every single assistant professor to apply for the biotechnology category also. The reason is that some universities, they have combined both biochemistry and biotechnology in their universities. So if you want to apply for the biotechnology background, if you want to apply for a biotechnology post, even though you are from biochemistry, you are eligible. If you want to apply for biochemistry, even though you are from a biotechnology background, you are eligible in those specific universities. That's why I'm requesting all the assistant professors, if you are applying for biochemistry, please apply to the biotechnology. If you are applying for the biotechnology, please apply for the biochemistry as well. And I will do the comparison of syllabus also, because friends, 60 to 65% of the biotechnology and biochemistry syllabus is almost the same. Hence it's a great advantage to all of you. Okay, now let's start the detailed analysis. Requesting all of you watch the video till the end, then you'll get a complete idea. Now the point is here, there are 12 units are there. Now if you ask me how many number of units that we have to cover, every student know every aspirant know very well, we are having 150 questions in the question paper. You know it very well. And each question carries 3 marks. That also you know very well. So you have 150 questions. How many units are there? 12 units are there. So out of 12 units, from every single unit, there is a no doubt. There is a no doubt. You can expect 12 to 13 questions an average from every single unit. Every single unit. Hence, in order to score out of 150 questions, trust my words blindly, if you answered, I'm talking especially for the general category, 100 to 105. If you belong to the BC, ST, SC category, if you answer at least 90 to 95 questions, general category 100 to 105 questions, remaining other categories, if you scored 90, if you answer 90 to 95 questions for sure, trust my words blindly, you can very surely crack the exam. You can reach yourself up to the interview. Right. Now, the second and most important one, how many units has to be studied? Out of 12 units, I will tell you what units to be focused. 9 units. 9 units you have to read 100%. I will tell you what are those 9 units that you have to study 100%. Then what, either 1 or 2 units you have to study 50 to 60%. That is sufficient. So what I'm trying to explain to you, you have to focus on 9 units completely. 1 or 2 units, that is the 10th or 11th units, you have to read 50 to 60%. Then undoubtedly, friends, we can crack the exam. Now let's focus. What are those 9 units that we should not skip under any circumstances? Unit 1, I will do a detailed analysis, don't worry. Unit 1, chemistry of biomolecules, bioanalytical techniques. Unit 2, enzymology. Unit 3, and cell biology, bioenergetic, human nutrition. In this, I will tell you what you can avoid in the unit 4. Unit 5, compulsory intermediary metabolism. Unit 6, microbiology and microbial genetics. Unit 7, very important, molecular biology. Unit 8, immunology. So friends, I don't encourage any student, including the molecular physiology. These 9 units, I don't want any aspirant to leave it. I will focus on what you people has to read. Please don't worry about that. Then, when it comes to the unit 10, unit 11, unit 12, I am requesting all of you in the unit 10, unit 11, unit 12, you choose any 2 units according to your understanding, means how depth you already know the concepts of this 9, 10, 11, 12 units. Out of this, pick 2 units. I will give my suggestion as well. And most importantly, in this 50 to 60%, if you cover, that's more than sufficient. We can crack the exam. Okay, let me explain you one by one unit. Now, the assistant professors who are already working in the biochemistry department from years together, who are having 10 to 15 years of experience, I know some people will feel the syllabus is intermediary. Most of the assistant professors who are already working and the students who just completed their MSc and who qualified set or net and who is writing this particular exam, they feel little more difficulty. The reason is that the syllabus is extremely vast, very vast syllabus. And moreover, in your MSc level, even assistant professors, majorly we use it to focus on theory. When I'm not here to criticize anyone, I'm just telling you the overview. Most of the assistant professors are continuously focusing on the theory wise in our MSc teaching. But when it comes to this exam, all the questions will be application based. all the questions are analytical, all the questions are numerical, all the questions are graphical, all the questions are factual statement based. So you people require a wonderful application knowledge of every single concept. Then only we can answer the questions. Okay, now let's see. Let's start with the chemistry of biomolecule. So friends, here name itself indicating, right? We have to focus on the chemistry of four major biomolecules, starting with amino acid, protein, lipids, as well as the nucleic acids, followed by the polysaccharide as well. Now, I can tell you very clearly among this particular unit one, amino acid, proteins, lipids, nucleic acid, where you people can get the numerical type of questions. First point, in case of a amino acids, you people will get a question from pH related numerical, pKa related numerical, isoelectric point related numerical. In order to make the concept more difficult, in order to make the paper more difficult, they may raise the numerical question even from the Ramachandran plot also. So you people has to focus on very simple classification of all the 20 amino acid with respect to their structure, function, basic amino acid, acidic amino acid based on their R group, all the type of classification we have to focus on, followed by the pH calculations and pKa calculation. I'll explain you very depth in the classes. Please don't worry about that. And what about the proteins? We have to read the complete protein classification, including structural also, functional also, as well as characterization of proteins. And friends, this is not a difficult part. This is pure theory part. You can do it very easily. When it comes to the classification and chemical properties of carbohydrates, friends, this is a detailed analysis class. I'm only explaining you the what to focus for the exam point of. I am not explaining you the lessons. For this, we should majorly focus on the linkages. What do you mean by that? So what is the major linkages of the polysaccharides, such as cellulose? What will be the glycosidic linkage? What will be the glycosidic linkage in the inulin? What will be the glycosidic linkage in the chondroitin sulfide? What will be the glycosidic linkage in the heparin sulfate? Which are the major polysaccharides that we have to focus on, because they have very clearly mentioned in the syllabus heteropolysaccharide major ones, glycosaminoglycans and glycoproteins. These three are the major type of the polysaccharide where we can expect the questions from the linkages, which we have to focus on, followed by the disaccharides like lactose, maltose, sucrose. Those linkages also we have to focus on. Then next one, classification of the lipids, fatty acids, and their physical properties. Trust me, friends, here also you don't get the questions from the linkages. There is a possibility that they'll give the fatty acid, they may give the structure, and they will ask us what is the chemical name of that particular fatty acid. Otherwise, they will raise the question from the classification also. Otherwise, they will ask the questions, the differences between the plant lipids as well as the bacterial lipids. Otherwise, they will ask the question, which of the following lipids are abundantly found in the nervous system? They can ask the question, which of the following lipids are most abundant on the plasma membrane? Which of the following lipids are most abundant in the mitochondrial as well as the chloroplast membrane? Trust me, here also you are going to get purely theoretical only. There's no need to worry about it. When it comes to the nucleic acid, friends, I want all the students give the highest importance to the DNA when compared to the RNA. Because friends, when we are reading the DNA, they majorly focus on the types of the DNA. That is the A DNA, B DNA, Z DNA. Especially they raise the questions from the differences. That is, one turn of aDNA contain how many number of nucleotides, what is the pitch length, and what is the sugar puckering and base conformations. Especially they will raise the questions from the differences between the one type of DNA to the another type of DNA. And when it comes to the RNA, friends, they will majorly focus on their synthesis. where the tRNA gets synthesized, where the mRNA gets synthesized, where the rRNA is getting synthesized, and their location, which enzymes are involved, what cofactors are involved. Those kind of questions they will ask from the nucleic acid. It's not difficult. It is also easy. Most of the assistant professors are already teaching from many years together. I strongly believe that this kind of small concepts will be in your fingertips. Then porphyrins. When you are reading the porphyrins, this we can include with the plant physiology also. Structure and properties of porphyrin, heme, chlorophyll, and cytochrome. Right? Suppose example, if you take a chlorophyll having a porphyrin head, is it? And chlorophyll having a phytol chain. So we have to just understand what is the structure of a porphyrin, what is the structure of a phytol, and what is their composition and what is their functions. Especially they have mentioned only one type of a photosynthetic pigment, only one type of a pigment that is involved in the sensory photobiology. That is the cytochrome. It is very easy to understand. I'll explain this in just one class. So under any circumstances, you should not miss the unit 1. Next one, unit 2, bioanalytical techniques. Now you can ask me, sir, what kind of questions that we can expect in the bioanalytical techniques? Every student remember my words. In the bioanalytical techniques, you can expect the questions. Remember, numerical, you can expect from graphical, you can expect from statement related. There is a possibility you can also expect the questions from the matching as well. Now, it's a very vast syllabus in unit 2 also I'm saying, but I will focus on where you will expect what kind of questions. And when it comes to the electrode and indicator dye technique, we are going to get the questions either from the pH measurement. If not from the pH measurement, there is a possibility that from HHB equation, you may get the numerical Henderson-Hasselbalch equation. But trust my words, you will get a very basic level numerical questions only. You don't get any depth of the questions. Then where we will get a difficult questions from the unit 2, all of you remember, you are going to get it too much difficult questions from the techniques. Now you can ask me, there are multiple techniques were given in the exam. Is it really worth of reading this many techniques at this specific point of time? No. So I'm marking it. Trust my words, the questions are going to appear only from those techniques. GLC, HPLC, IR exchange chromatography. These three are very fundamental in case of chromatography technique. And I am requesting all the assistant professors to give a highest importance to the spectroscopic technique. Start from UV visible, IR, no need of ESR, focus on the NMR, no need of ORD, focus on the CD, focus on the X-ray, focus on the mass. So friends, UV visible, IR, NMR, CD, X-ray, mass. This is 6 spectroscopic techniques under any circumstances. Every one of you has to read it. And when it comes to the radioactivity, I encourage to undergo the overview of this entire radioactivity. I'll explain this entire radioactivity in one PPT in one class. That will be more than sufficient. Friends, when it comes to the electrophoresis, again, focus on the SDS phase, focus on the capillary electrophoresis, isoelectric focusing, and two-dimensional electrophoresis. Pulse field, you just try to understand the application of this. Is not greatly important. So focus on these 4 type of electrophoresis techniques. Blotting technique. Blotting is very important, and most of you people having a wonderful idea about all the blotting techniques because you might did many experiments throughout the years. Hence, you are very good in the blotting techniques. I encourage all the assistant professors to focus on all the type of blotting techniques. In our exam, there is a possibility that we can expect the questions from the matching. So I am requesting every one of you to focus on the application, especially application based. Okay. Now, when it comes to the biostatistics, now you can ask me what kind of questions you can expect. I can tell you there is a possibility of matching question. There is a possibility of the numerical question, mostly numerical, especially from the mean median mode. Right. If not, there is a possibility that you can get the question from the regression lines. It is not that much difficult, friends. With a simple formulas, you can calculate this particular question. But since you are all having a negative marking, until you are very sure about your answer, I don't encourage any assistant professor not to go through the numerical part in the exam. But in the class, I will teach you simple shortcuts, how to focus on this particular numerical problems. Please don't worry about it. So that is about unit 2. When it comes to the unit 3, enzymology, again, here you will get more graphical, here you will get more numerical, here you will get more statement related, here you will get more matching also. Because you also know very well, it is a very vast unit, very vast. You know, in your syllabus, in the MSc first year and MSc second year, whatever you use it to teach, from years together, they just copy pasted the entire syllabus here in the enzymology category. Then you can ask me, sir, what we have to focus on? See, let's look into the first subunit. This is all historical aspects. Here, you can get the questions majorly from the examples, which means that what are the different examples of the coenzymes? What are the different examples of the cofactors? In other way, which suppose example, let's take zinc. It is acting as a cofactor for which enzyme? Molybdenum acting as a cofactor for which enzyme? Manganese acting as a cofactor for which enzyme? Arginine is acting as a, silver acting as a cofactor for which enzyme? These kind of questions that we can expect from here, which are majorly for the example wide, and structure of the active site, and various models to explain the activity of the enzyme and substrate relation, that is the Fisher and Coastland models, formation of the enzyme substrate complex formation, most importantly the factors affecting the enzyme and substrate complex, that is substrate concentration, environmental factors, pH, temperature, all these very basic level questions only you can expect from here, and where you will get a pure numerical questions, undoubtedly from the kinetics. Trust my words, friends, especially from the kinetics only, you can expect minimum 3 to 5 questions. And I will tell you, friends, which kind of numericals that you have to focus on. Give the highest importance to the Michael-Menten equation, followed by Lineweaver-Burke plot, AD-Hofstee, Hans plot. These 4 you have to give the highest importance. And you have to know the significance of Vmax. You have to know the significance of Km. You have to know especially the significance of the Kcat by Km. And most importantly, how to calculate Vmax, how to calculate Km, how to calculate Kcat by Km. These are also important with respect to the numerical types. I'll explain in a very simple manner in the exam, in our classes. After listening my classes, you can answer the questions very, very easily. If you did not get the questions from these equations, again, there is a possibility, friends, we will get the questions in the form of graphical or numerical or statement related or equation based also. You will get from the inhibition. Especially focus on reverse, non-competitive. Enough. There is no need to read partial, substrate, but focus on the allosteric. That is very, very important. No need of feedback also. Focus on the reversible, that is competitive, non-competitive, allosteric. These 3 are very important. I want you to focus on that. Then this third subunit, third part and fourth part, this is also again pure theory. Pure theory. In this third subpart and fourth subpart, I will request everyone to focus majorly on the fourth part. Why? Because from the regulation, we can expect a very good type of questions. In the CSR exam also, even in the degree lecturer exam also, we used to get a more number of questions from the regulations only. Hence, I encourage every one of you to focus on the regulatory concept compulsory. Especially, I will in the classes also, in the PPT also, I will tell you what to focus on. Give the highest importance to the Hill equation plot, and don't focus on all the enzyme complexes. Focus on glycogen phosphorylase glutamine synthase. Pyruvate dehydrogenase complex, which is a widely asked question in many exams multiple times. Don't worry, whenever I am teaching, I will stress very specifically. You people focus only on those specific units. Don't worry about that. So unit 4, I can say that all the questions from the either statement or matching. You don't get any kind of a difficult questions. I mean to say you will get a difficult question, but not any type of a numerical or graphical questions. Here, what is this cell biology, bioenergetic and human nutrition? This is a kind of a unit where they combine the entire cell biology, a little part of human nutrition, they combine the entire bioenergetics. Now, which is very difficult here, difficult in the sense where we can expect more number of questions. We can expect more number of questions from cell biology compared to the human nutrition and bioenergetics. You all are reading the cell biology and you are teaching to the cell biology to the students years together. And I want you to focus on, I will teach a very depth. You don't need to worry about it. You focus on the structure of an organelle, function of an organelle, enzymes present in that particular organelle, deficiency, diseases caused by the deficiency of a specific enzyme. Most important for everything, protein sorting. Don't worry, I will tell to every one of you what are the organelles that you have to give the highest importance whenever I am teaching you the lesson. Then, where you can get a very difficult question, friends, you will get from the question from cell cycle. Again, I am telling to all of you, you don't get any kind of a questions from what is mitosis. You don't get any kind of a question from what is meiosis, their significance. Here, they will exclusively focus on regulation, which means that you will get the question from what kind of cyclins and what kind of CDKs that are regulating the G1 phase, S phase, G2 phase, M phase. 99% of the cases, in order to make the question very difficult, they will combine the cell cycle with the cancer and apoptosis. They will raise the questions. They are little tricky, but if you know the depth of the concept, friends, you can answer that particular kind of questions very easily. Don't worry about that. Then bioenergetic composition of food and vitamins and minerals. And even whenever I'm teaching, I really don't focus on this vitamin minerals composition of food, because it is just basic BSc level syllabus, a basic MSc level syllabus. Everything is very straightforward. And here entire these two small subunits are completely memory based. I don't want to focus on the concepts which are memory based, which you people can remember if you read 2 days also. It's very easy. Hence, I am requesting all of you, I want you to focus on these two. Anyway, I'll provide the PPT, everything. Don't worry about it. But even though I teach, I may not teach, you can study it very easily. When it comes to the bioenergetics, again, it's also very important concept where you can get the numerical questions, especially from the entropy related, enthalpy related, even Gibbs free energy also, you will expect the numerical question. This we will discuss very elaboratively. And friends, in the bioenergetics, very commonly where we can expect the questions. Listen, all of you look into this. Classes of electron transferring enzymes, and trust my words, you are going to get the question damn shortly, very surely, from the inhibitors that are blocking the electron transport chain. So if you look into the mitochondria, there are four electron acceptor complexes are there. 4 electron acceptor complexes that can be blocked by a different type of inhibitors. We will expect a matching question. We will get a experimental based question also. Suppose example, a student is performing a mitochondrial experiment. As a part of experiment, they have used some inhibitors. That inhibitors blocked certain electron flow. Then they will ask what is the mechanism of that particular inhibitor, or they will ask that inhibitor blocked which electron system. This kind of questions that we can expect. And here only they have included cyclic and non-cyclic reactions of photosynthesis. They have included photorespiration or the C2 cycle. It is very easy but little tricky. But don't worry, I'll explain you very depth. Now, Unit 5 and Unit 6 are too much important. And I can tell you, friends, why the Unit 5 is extremely important, because the syllabus is vast. And trust me, friends, you will expect too much, to say more number of questions. Let's talk among all in the Unit 5. The most important one is the intermediary metabolism. This subunit is very important. Why? Because you see this, they have mentioned glycolysis, they have given glucogenesis, sorry, they have given gluconeogenesis, they have given glycogenesis, they have given glycogenolysis, they have given PPP pathway. And see this, in order to make your concept very difficult, they have especially asked the enzymatic activity also. They focused on the glycogen phosphorylase enzyme, which is involved in the breakdown of glycogen. They focused on glycogen synthase enzyme, which is involved in the glycogen synthesis. Here also you will get a very tricky questions, which they use to ask in the CSR also. Now you can tell me, we all read these kind of equations, pathways years together. Trust me, you are not going to get the equations directly from the names. You are going to get the questions from the regulation. Which are major regulatory enzymes in the glycolysis? Which are major regulatory enzyme in the TCA cycle, including their structures, how they are going to get regulated, which are acting as a positive regulators, which are acting as a negative regulators. That kind of questions makes the paper little more difficult. But trust me, with a thorough understanding of the concept, you can crack the question. Then metabolism of fructose, galactose, and lactose. This is easy, but there is a possibility that we will get a less number of questions from here. Why? Because they majorly focus on the intermediary metabolism when compared to the monosaccharide, disaccharide, and polysaccharide. Right. So then give the highest importance here. Then see here, protein turnover. See, and one more important point is that in your syllabus, luckily they have mentioned where to focus on. This is actually a subunit which is combining the human physiology and biochemistry. Right. In this, I am requesting all the assistant professors give the highest importance to the urea cycle, and I am requesting every one of you focus on the amino acid which are having the neurotransmitter activity. That is more important for the exam. Focus on the regulation of a branched chain amino acid as well as aromatic amino acid. Don't worry, I'll explain you very depth in the exam. We will focus very specifically the important amino acid that are acting as a neurotransmitter, which are very important towards the exam point of view. We will study on it. Now, fats as energy store, oxidation of fatty acid. And trust me, friends, this is also too much important. Here, when we are studying about the oxidation of the fatty acid, remember my words, give the highest importance to the beta oxidation, because from here only you are going to get the question. After the beta oxidation, give the highest importance to the cholesterol metabolism. There is no necessity to read anything else except these two, beta oxidation and cholesterol metabolism. I'll cover this within the 2 classes. Then whenever you are reading the nucleic acid, again give the highest importance to the Salvage pathway, which is very important. So for the DNA synthesis, we have only 2 pathways. One is called as the de novo synthesis pathway, Salvage pathway. If you focus on these two along with their skeletal structure, that's enough. You can crack all the questions that are coming from here. Easy unit and you should not miss. Then microbiology and microbial genetics. This is also too big. Now you see this. They have combined the genetics and they have combined the microbiology. I'll tell you where you are going to get a difficult question. Let's consider the scope of microbiology. Here you are going to get a very, very basic level question, especially from the cultures, structure of the bacterium, very important, friends, then followed by the classification. Trust me, you're going to get a very direct question. Same with the viruses also, because classification, suppose example I'm telling you, They will give all the viruses in one column. In other column, they will ask whether they are having a DNA as their genetic material or RNA as their genetic material. They will ask which virus having a single-stranded RNA as their genetic material, which virus having a double-stranded RNA as their genetic material. Here we get the questions very straightforward, informative. If you know the answer, you can answer. If you don't know the answer, you cannot answer. Straightforward questions only. Now, when it comes to the cell growth and kinetics, trust me, you're gonna get the numerical or statement or matching. Many times they use it to ask the matching questions from the nutritional requirement. Not only nutritional requirement, they will ask the question, which are the following are example of aerobic bacteria, anaerobic bacteria, oxygenic, anoxygenic, which are phototropic, which are mixotropic, which are chemotropic, right? Which are pH dependent, which are temperature dependent. These kind of questions that we use to get straightforward, informative based. It is not at all very difficult questions. In growth and kinetics, especially from the lag phase, log phase, stationary phase, as well as decline or death phase, you may expect the questions which are the numerical type, either numerical or the statement related or the matching. But trust me, it is easy. It's not difficult. And it's a good subunit that we have to focus on compulsory. Now, when it comes to the genome organization and recombination, so suppose let's focus on the recombination. Trust my words. All the assistant professors, I am requesting you, you have to give a highest importance to the transformation, conjugation, and transduction. Trust me, you are going to get only numerical. Apart from the numerical, you are not going to get any other type of a question. And trust me, this kind of numerical questions are also extremely difficult. You require a very good understanding of the concept. Then only you are going to answer that questions. Otherwise, it's very difficult to answer these questions, because most of the questions are related to the mapping. But trust me, if you attend my classes, you can answer 90% of the questions are very easily from this particular thing. And genome organization, this is not that much difficult. This is not that much difficult because mostly theory based questions only you will get. Especially we will get the questions from the eukaryotic chromosomes only. That you please don't worry. I will cover this entire genome organization within 3 classes very elaboratively. It's very straightforward because all the questions from theories only. It is easy. Then the unit no student should not skip is the molecular biology as well as the immunology. If you ask me a simple question, sir, should I leave anything? I will tell to all the students, 100% unit 7 and unit 8, you have to read. You know, if you ask me a single question, is there any important concepts that we have to focus on? No, I can't say that important because that much depth you have to read. I will just overview in a simple line. You have to focus on rules. You have to focus on enzymes. You have to focus on process. Every student listen. You have to focus on rules. You have to focus on enzyme. You have to focus on process of replication, transcription, translation, post-transcription, post-translation, gene regulation in both prokaryotes and eukaryotes. In addition, what they mentioned? In addition, they have given the DNA repair mechanism. See, friends, if you want me to explain the analysis of this entire unit, it will take a lot of time. I can tell you in simple words, whatever you assistant professors are teaching in MSc first year and second year, those things we have to read exclusively application based. I don't encourage any one of you to leave any concept from unit 7 molecular biology and unit 8 immunology. But trust me, friends, if you read my lessons, if you attend my classes, trust me, you can answer any kind of a question from molecular biology, any kind of a question from immunology. Do you really think you have a time of reading the entire Watson book of molecular biology? Do you really think, do you have a complete, you have a time to read the complete QB by immunology? You can't. But whatever the PowerPoint presentations that I will prepare from all the standard reference book, not 1 or 2, more than 2 or 3. Hence, if you follow my PPT and if you follow my books, You can read any kind of, you can answer any kind of a question. So what I am explaining to all of you is, you no need to worry about this. I will focus what questions that we will get in the exam. You can answer any kind of a question if you strictly follow my lessons. Don't worry about this. And in the molecular biology and immunology, I don't tell you that these units only, these points only we have to focus. We have to focus 100%. Every point, every pinpoint we have to focus on. Hence, I'll teach in a very detailed manner. Don't worry about that. Then again, molecular physiology. This is a type of a unit. You know what? See, I don't know how they combined it. They have combined cell signaling only here, and they have combined cell communication here. They have combined a cell membrane here. They have combined a blood vascular system here. They have combined muscle mechanisms here. So different things they have combined. Now, if you ask me, sir, in the entire molecular physiology, which is the most important one? I can tell to all of you, undoubtedly cell signaling. And to be specific, friends, compared to the CSR syllabus, this syllabus is little easy only. Well, little easy, because they have not mentioned that much depth of the cell signaling. Right? I will explain this within 4 hours of classes. We will do in a detailed analysis. Now, the composition and structure of the cell membrane, it is easy. We will study in the cell biology itself. Then structure and organization of the muscle cells. This is little rigid topic, and the point is that you all people already studied, and you are all teaching from years together, and you will get a theory based questions only. From the cardiovascular system, blood circulation, muscle movement, nervous system, you are going to get a very direct questions. You get a little tough questions from the composition of cell membrane and cell signaling. It is a small unit. We can cover very faster. Then clinical biochemistry and endocrinology. I can tell one point to all of you. You will get more questions from the diseases. More questions. Deficiency diseases. And you will get the questions from the tests. Liver function test, pancreatic function test. Because the people will majorly focus on the clinical aspects here. What they will focus on? They will majorly focus on the clinical aspects. Hence, this is not difficult, friends. This is not at all difficult. See, because see this entire endocrine system, they have broadly, I mean to say, differentiated. They asked us to focus on specifically kidney, focus on specifically pancreatic hormone, thyroid hormone, adrenal hormones. Their secretions, simple secretions or the regulations, functions, diseases, tests. That's all. Is it really difficult? No, but it is a rigid topic because you have to focus on maximum amount of information. Hence, it's very important that you have to give importance to remembering only or memory based only. That I'll help you. Don't worry about that. Then genetic engineering. I don't encourage you to leave this genetic engineering because I'll tell you, in the genetic engineering, you focus on complete rDNA technology, which means that all the enzymes involved in the rDNA technology, all the vectors involved in the rDNA technology, host involved in the rDNA technology, methods of transformation, followed by the techniques. This entire genetic engineering, this part is a very crucial part. That is the cloning vector, followed by technique like a PCR, RT-PCR, and luck is that they have mentioned the specific techniques only. Hence, we can focus on the specific technique instead of focusing on everything. And this entire unit also, I can finish it in 6 to 8 hours, very, very elaboratively. Don't worry about it. Then unit 12, I already told to all of you, if you want to leave it, leave. If you want to focus on, focus. I don't worry about it because we don't have that much of time. Okay. And don't worry, I will start one more session. I'll do one more session according to the subject wise also. And remember, from November 10th onwards, the classes will be commencing. I will give you details. Don't worry about that. The timings will be because most of the assistant professors you are already going to the universities. So for your convenience, I'm keeping the time 5:30 to 7 a.m. and evening 7 to 10 p.m. And trust me, if you are very, 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 very dedicated, if you are very, very dedicated, and if you want to crack the exam, very surely then only consult me. I'm requesting every one of you, don't consult me just for information. If you want information, you can get the information anywhere. If you are very dedicated, you want to crack assistant professor and you want to settle in your life, then only contact me. Please don't waste your and my time. Okay. If you are dedicated, then only I will help you in every part of your preparation, every step of your preparation. And I am very sure that if you follow my classes, 100% you are going to crack the exam. My study material, MCQ series will help you a lot and lot to crack the exam. If you are joining with me, I am expecting only two important characteristics. One is your patience, and most important one, your interest. If you are having these two, I am there to help you. 100% you will crack the exam, and it's my word. Okay. Thank you. If you have any kind of a doubts, you can WhatsApp or you can call on this number. Okay. Thank you. Thank you so much. Thank you.